Now we're going to move on to transcription, which is making a short copy of DNA, and where that copy is going to be made of RNA, and it's going to be used eventually to make some protein. So, the first thing that's going to happen is we're going to have an enzyme set down at the beginning of the gene, and this is RNA polymerase. Now this is like primase, but instead of just making a primer, it's going to make a copy of this entire gene. And the first thing that it's going to do is it's going to break these hydrogen bonds between the nucleotides, the two strands of DNA, and it's going to unwind the helix. Now, that's what helicase does during DNA replication. But RNA polymerase, because it only has a short stretch to do instead of the entire genome, which may be millions, if not billions, of base pairs long, what it's going to do is it's just going to do this short stretch. So not only is it going to act like helicase and unwind the helix and break these bonds, it's also going to put an RNA copy, or it's going to be an RNA polymerase for this template strand right through here, the blue one. And then that copy is going to go off and be involved in transcription. So I'm going to pause the video while I write in this copy and this unwound segment of DNA. So I show these strands as being separated already, and that's just to save time while I'm drawing. But the RNA polymerase is going to move this direction and break these hydrogen bonds and unwind the strands. Now, it's going to make a copy of just this bottom one, and it's going to go from 5' to 3'. So, what goes with TA? Now, remember, this is RNA, so we don't have a T in RNA, so we've got to make a U, and then we're going to make a G, and then I'm going to keep going along, C, U, A, U, G. And as this goes along, this is going to start falling off as the RNA polymerase moves along. So I'm going to go ahead and put a few more nucleotides on. So U, A, G, G, U, A, G, G, and I'm going to erase this, and I'm going to show this falling off of the template strand. So A, U, G, C, U, A, U, C. See, so it's kind of falling off. So eventually, I'm going to have an mRNA, and I'm going to write this in black. And M stands for messenger. So I'm going to have AUG, CUA, AUG. There should be a G up here. UAG, GUA, AGG. I didn't leave myself enough room, so I'm going to have to go around the corner and UAA. Okay, so that's transcription. I have made an RNA copy of the DNA template or the DNA gene. So for extra credit, where is this happening in the prokaryotic cell? This is happening in the nucleoid region.